Welcome back to this next video in the series of lectures on the cell cycle. In the last video, I've told you that the E to F transcription factor that was released from the retinoblastoma protein by the action of the G1-CDK, and the E to F was actually going for the transcription of a cyclin that was increasing the level of the cyclins in the cell, and the cyclin E was interacting with cyclin dependent kinase 2, making the G1-S-CDK. Now the G1-S-CDK, what it was going to do, that it was converting the pre-replicative complex into the pre-initiation complex by addition of multiple proteins to this pre-replicative complex, and one of the important protein that was the CDC45. This G1-S-CDK, it was also going for the phosphorylation of the P20 protein, thereby promoting the transcription of the histone genes. The G1-S-CDK was also going for the initiation of the centrosome duplication. Another important function of the E2F was to release an inhibitor from the promoter region of another cyclin, which was the cyclin A, thereby increasing the concentration of the cyclin A in the cell. This cyclin A was then going to, that was actually interacting with the CDK2, the cyclin dependent kinase 2, making the S-CDK. And this S-CDK was actually ensuring that the replication of the DNA that goes well, and it was also ensuring that the replication of the DNA is only once per cell cycle. Now, how the S-CDK was going to achieve that it was going for the phosphorylation of the origin recognition complex, and by phosphorylating the origin recognition complex, it was actually making the environment unfavorable for the attachment of the CDC6 protein. The S-CDK was also going for the phosphorylation of the CDC6 protein, thereby making it for a degradation by the proteasome. The CDT1 protein that was actually inhibited by another protein, which was the germanin, and the level of the germanin that was actually increased by the activation of the E2F protein. So this was the mechanism by which the S-CDK was actually ensuring that the during one cell cycle the DNA is going to replicate only once. Now in this video I want to talk about the S phase of the cell cycle. Now in the S phase of the cell cycle, you are going for the replication of the DNA. So all of your DNA, all of the DNA molecule in the cell, they are going to get replicated. This is single, I'm only talking about a single chromosome, and of course these mechanisms apply to all of the chromosome or the DNA molecule that are present in the cell. When we talk about the diploid organism like the human beings, they have got the homologous chromosome. The chromosomes, they are in pair, and that pair of the chromosome is known as the homologous chromosome. And in this pair, you are getting one copy from your mother and you are getting one copy from your father. During the S phase of the cell cycle, what you are going to do is that you are making identical copies of both of these chromosomes. You are actually making an identical copy of the mother's DNA molecule. You are making an identical copy of the father's DNA molecule. And when you make the identical copies, these identical copies, they are known as the sister chromatids. So this is one pair of the sister chromatid. This is another pair of the sister chromatids. Because they are identical to each other, therefore we call them as the sister chromatids. Now these sister chromatids, they are attached to each other by a protein complex that is known as the cohesions. And these cohesions, they are attached after the DNA that has been replicated, and you have got the sister chromatids. Now by attachment I mean that these two sister chromatids, they are attached to each other throughout their length by these cohesion proteins, and this pair of the sister chromatids, they are attached to each other throughout their length by these cohesion proteins. If you look at the functions of these cohesion proteins, they are actually responsible for the regulation of separation of sister chromatids during the cell cycle. What I mean by that is that these cohesion proteins, they are attached until the NF phase of the cell cycle to these sister chromatids, and they are working as a checkpoint. By that I mean the checkpoint means that they are actually regulating the separation of the sister chromatids during the cell cycle. We will discuss that in detail when we will be discussing the anaphase of the M phase of the cell cycle, that how these cohesion proteins are acting as a checkpoint, thereby ensuring that the separation of the sister chromatids will be highly regulated, and both of the daughter cell, they will be getting equal amount of the genetic material after the cells have divided. These cohesion proteins, they also have some other functions, and the important functions are the regulation of the transcription, and they are also responsible for the DNA double strand break repair. As this cohesion is a protein complex, so if you look at the composition of this cohesion complex, they are having some important proteins. One of the important proteins, they are known as the SMC1 and SMC3. Now this S is for structural, this M is for maintenance, and this C is for the chromosomes. So they are the structural maintenance of chromosome 1 and the structural maintenance of chromosome 3. These are the two important proteins. They are making up the coherence. Another important proteins that you find in the cohesion complex are the SCC1 and the SCC3, which are actually the sister chromatid cohesion 1 and the sister chromatid cohesion 3. So this is actually the proteins. They are responsible for making up this cohesion complex, which is ensuring the equal distribution of the sister chromatid between the two daughter cells. Once you have replicated your DNA, you have attached the cohesion proteins to all of the pairs of the sister chromatids. You are actually moving from the S phase into the G2 phase of the cell cycle. Now, this G2 phase of the cell cycle, during this phase, the cell is growing very rapidly. They are making a lot of proteins. They are doubling their organelles. They are going for the duplication of their organelles as well. And actually the cell is preparing itself to move into the M phase of the cell cycle. Now this G2 phase, in some of the literature you will see that this is actually the quality control phase. What I mean by that quality control phase, if you have ever visited an industry, you will see that they have got two major units. One is the production unit and the other one is the quality control unit. Now when the production unit, they produce something, the function of this quality control department is to check whether the product has been made up to the standards or it have got some problems. So once the quality control division, they passes the product, that you can actually use that product for the consumers, and you can market that. Now if you look at the cell cycle, this G2 phase is actually working as a quality control phase, because in the S phase, which is before the G2 phase of the cell cycle, you have replicated the DNA, you have made a product. Now you have to make it sure that the DNA product that you have made is complete and that is intact, no damage is there. This is what the G2 phase, the quality control phase, means. Now, how this G2 phase is working as a quality control phase then? It is actually going to check for the replication forks, so as to ensure that the replication of the DNA that is complete. Because the presence of the replication forks mean you have not completed the DNA replication yet, you cannot move from the G2 into the M phase of the cell cycle. So if there are no replication forks, that can be one of the signal that the DNA has been completely replicated. You can move from the G2 into the M phase of the cell cycle. Another important thing is that in the G2 phase, we have got this checkpoint known as the G2M transition checkpoint. Now this checkpoint is ensuring that the DNA has been completely replicated, and it's also checking that the DNA is intact. Now if there is any damage to the DNA, that means you cannot divide the cell with damaged DNA, because that will be leading to the formation of the abnormal cells. So when there is any DNA damage, the p53 is going to get activated. It is going for the transcription and for the synthesis of a protein known as the 14:3:3 sigma that is going to halt the cell cycle at the G to M transition point until you have repaired the DNA or you have taken the decision for the apoptosis. We will continue this discussion in the next video.